All right, everyone. Welcome to Foresight Molecular Machines Group. Really, really happy to be starting into a new year with you guys. This group is now in its, I think like really third year iteration. So it's really exciting. And I think we're, definitely picking up steam, growing. The applications are really keeping coming. So thank you so much for everyone for nominating such wonderful participants and for nominating such great speakers. We're really great for Q1. And I'm really excited for our upcoming workshop as well. So maybe just to bring people up to speed who may or may not know exactly what we have awaiting for you, we have, in terms of our seminars, then Molecular Nanotech ones can always be found on our website. We have Chris Schafmeister today. So he's already up on the schedule. And we had lots of really wonderful seminar presentations in the past. Feel free to dive into them. They all have a seminar summary. But if you're interested in what we have in store for you in the rest of Q1, you can just click up here. If you are interested, if you're watching this on YouTube and you want to join, and this is the application button. And then the second thing that I want to make you aware of is our molecular systems and design workshop that's coming up. We're already starting to plan it. We have lots of really wonderful folks joining us already. So if you want to meet Chris in person, if you get inspired by his talk today, then that's a really great way to do so. And so please already start applying, and we're definitely accepting applications on a rolling basis already. And so those are the two major announcements. If we have time, maybe I'll introduce you to the fellows after the talk. But for now, really, really excited to have Chris Schafmeister join, who's definitely been a very long-term friend of Foresight's. And, I think usually whenever we had our previous seminar reports written, it was always straight to your section that many people skipped. And that's been really exciting. So thanks a lot for coming on. And you're at Temple University. You'll be discussing candidate hardware and software of a critical path to bottom-up molecular nanotech, and hopefully perhaps also some bits and pieces about third law in there. So I'll be in the chat. I'll be monitoring questions. And then I'll be funneling them to you later on. So feel free to already keep them coming as Chris is starting to prep, and I will gobble them up and then relay them later. Okay, wonderful. Well, the stage is yours. Thanks a lot for coming and really excited for the talk.
Okay, thank you for the introduction. Can everybody hear me?
Loud and clear.
Okay, you can hear me? Great. All right, let us try. I think that'll do it. Okay, is that full screen now? Great.
Yes.
Okay. Just get this down. Okay. So I'm going to tell you, I'm going to just whip through a bunch of things that we're working on and some proposals I have for things that I want to work on. And so this is a rough outline of the talk. First, I'm going to tell you how we're going to deal with future pandemics using our technology, how we're going to cure aging, how we're going to provide abundant green energy, and how we're going to bootstrap molecular nanotechnology. And I know it sounds like a lot. The first one is already supported. It has been supported since 2019 by the Department of Defense, the Defense Threat Reduction Agency. This is the organization that protects the United States, protects us from chemical, biological, radiological, and nuclear threats. They are funding us to the tune of about $12 million. And thought I would start with what we are doing there in order to sort of show you how the basics of how our chemistry works. Okay. All right. So I actually have two groups of people that are working at about 45 minutes apart physically. This is my lab at Temple University. These are my students, one of my postdocs. This picture was just taken a couple of days ago. They've, Yihwei up here in the upper left has developed a lot of the basic chemistry that's made the technology that we're developing at Third Law possible. And then this is my group at Third Law Molecular. Two of these people are from Temple University. They are all chemists and one programmer, and they are developing this technology that's funded by the Department of Defense. We have a board of directors. Basically, how this started is I went to the Department of Defense in 2019, and I said, I think we could develop a technology that could replace monoclonal antibodies in diagnostic tests. And we could rapidly develop these things if a new threat comes along, a new virus, which, they come along now and again. we could rapidly develop molecules that bind them and then turn them into diagnostic tests. So they said, great, but we don't get anything out of academic research, so we want you to start a company. So that's what we have done. It is more of a research organization currently funded by the Department of Defense. All right, so here's the basics of how spiroligomers work. We have developed these building blocks. They are rings, and they have on them 2 amino acid groups. And for about 10 years, we figured out how to synthesize these things and how to link them together into larger structures. We made a few dozen of these molecules, determined their structures, but they didn't do much because they didn't have any functionality. They didn't have any side chains aside from the inherent structure of the core molecule. I'm just going to do a quick sound check. Can people hear me?
Yes.
Yes. Okay. All right. So then in 2010, we figured out how to get functional groups onto the building blocks so that every building block has a side chain. And then we figured out how to connect them together with these functional groups to create these ladder molecules that we call spiroligomers. Okay, you're probably all familiar with DNA, with proteins, peptides. These are different. The essential difference is that these building blocks link to each other through pairs of bonds. And so you get a complex, rigid, three-dimensional structure that you can decorate with all these different functional groups, and they get presented in 3D space in different directions. And then what we've been doing since 2019 is tying these molecules together to create really large molecules. These are about 5,000 Daltons. They're about the size of a small protein. And we have in the last 3 years made 2.5 million of these molecules and screened them against dangerous organisms given to us by the Department of Defense and identified molecules that bind those organisms. Just a quick summary. I'm not going to go through all this, but just a quick summary comparing spiroligomers to DNA, like DNA origami, versus proteins, with protein design, people designing proteins these days. Spiroligomers have some added advantages. For one thing, they're much more structured. They are ladder molecules, and we can link them together and then cross-link them to create little bundles that are where you can control basically where every atom is in three-dimensional space. They're extremely stable. They don't unfold like proteins and DNA does. We can put thousands of different functional groups on them, comparing to DNA, which has 4 chemical bases, and proteins, which have 20 amino acids in the natural suite of amino acids. We can put thousands of functional groups onto our building blocks. Spiroligomers are made now with robotic organic synthesis, and they're quite inexpensive to make. It's about $25 a gram for the monomers. And we could drop that cost to essentially zero with some technology I'll show you in a bit. Now, these molecules have a lot of applications, and we're really focused on developing applications. So the application I'm going to tell you first is this one, Symbas. And the idea is to link 3 of these together to make a molecule that's like a claw that can wrap around a protein and bind it and bury as much surface area as an antibody does. We've made 2.5 million of these molecules, and we've shown that we can rapidly identify ones that bind proteins. Another application is to tie them together into bundles with pockets and put metals down in the bottom of those pockets and create artificial enzymes, create catalysts that could do things like create synthesized solar fuels, cleave crosslinks that accumulate in our body as we age and potentially address diseases of aging. We can make molecules with pockets that could act as sensors for all sorts of specific small molecules like neurotoxins. We can make therapeutic molecules. We've made some molecules that bind proteins. And I believe that this technology will serve as the sort of a way to bootstrap molecular nanotechnology. So I'm going to show you some of these, how this works. Okay, so the first project is currently funded by the Department of Defense. We're about 3 years into it. We just passed another milestone last night. We've gotten about $9 million to date working on this thing. The idea is to make these really large claw-like molecules that can replace monoclonal antibodies and then incorporate them into lateral flow assays, like COVID tests that we've all been taking over the last couple of years, and develop tests for all sorts of different organisms that are indestructible, that don't have to be thrown out every 6 months, that you can warehouse for 10 years, pull it off a shelf, and they'll continue to work. And most importantly, that you can develop in just a couple of weeks, given the infectious agent, and start cranking out tests in months and millions of tests within like 6 months. That's the mission here. So this is a sort of basic idea of how this works. We've got here a white protein called trypsin. The red is the footprint of an antibody binding to trypsin. That's how much contact area you need to make to have one protein bind another protein well. This symbol here, this molecule with 3 segments, I've draped over it just to show a sense of the scale. And we're making very large molecules that can bury as much or more surface area than an antibody can. And that is essential to bind proteins selectively and tightly. So the first thing that we did that made this possible is we scaled up our synthesis. We contracted out the synthesis of a late stage intermediate of our building blocks, all 4 of these compounds, 1, 2, 3, and 4, to Wuxi Aptec. And they synthesized 12 kilograms of these 4 precursors in separate barrels. So it took them 6 months. We've got 4 barrels of these intermediates. We've used up about a third of it so far. And yeah, it's perfectly pure. What we do to it then is we further process it and we incorporate all these different chemical functional groups. These are all small pieces of known drugs. They're small polar heterocycles that are known to bind to proteins and are known to be safe as drugs, as part of drugs. So we display, we've got about 22 of them right now, and we make 2 forms of these. The core building block here has 2 stereocenters, and we make both the RR and the SS form of these building blocks. These are mirror images of each other. So we have 22 functional groups on 2 different backbones for a total of 44 different building blocks. This is just where we got started. There are thousands of groups that we can put onto these building blocks. Here's a picture of a few of this samples. On the left are the RR building blocks. On the right are their mirror images. And we have tens of grams of these. Then what we do is we randomly pick sequences, like 60 sequences for the week. We load up these robots with a couple of grams of each of the building blocks. And then every day we make 12 of these molecules here at the bottom. And the way this works is just like solid phase peptide synthesis. It's like robotic peptide or DNA synthesis. One building block is added to a resin, take off a protecting group, another one's added, take off a protecting group, another one's added, and you stitch together a molecule with whatever sequence you want. Then we hit it with an acid, which cleaves this from these plastic beads and takes off all these protecting groups on these groups, and then promotes a reaction where every building block attacks the previous one and makes a second set of amide bonds. This works beautifully. We have made 700 of these molecules in the last, in about 8 months. We make about 60 a week when we're running a production. We make 10 to 100 milligrams of each one of them. And each one of them has a specific sort of rod-like shape with different functional groups sticking off in different directions in space. And I've also developed software that lets us build 3-dimensional models of these molecules. And we are working now to get crystal structures of some of them. So then what we did is we take these molecules 3 at a time and we put them together in lots of different combinations. we make what's called a DNA encoded library. And we've made two different forms, these two different forms, each one of them containing about 885,000 members, where we've mixed and matched different segments in different combinations. So these are these claw-like molecules. And then we take these libraries of the compounds. They each they're on these small beads about the size of a human cell, 10 microns across. There's many copies of one of these, we call them SIMBAs, synthetic molecular binding agents. There's one version on each bead, and there's also a DNA tag that codes for the sequence. Then we mix it with a fluorescent protein. We mix 1,000,000 beads with a fluorescent protein, and the beads that have a molecule that binds that protein becomes fluorescent and lights up like a Christmas tree light. And the ones that don't bind the target are black. We then run it through a device called a fluorescence activated cell sorter, which sorts the fluorescent beads from the black ones. And you can shoot we currently run about 1.5 million beads through a day. And we isolate the beads that have bound to the protein. Then we go and PCR amplify up the DNA. We do high throughput sequencing of the hit beads using Illumina sequencing. And then we identify the hits that came up multiple times over multiple days, the real hits, and then we resynthesize them and we validate that they bind the target. So here's a bunch of beads that all display the same SIMBA, and they all light up because they've bound the fluorescent target organism. This is a bit preliminary, but we have done this 3 times now, and we've gotten hits in each case. And we have then taken some of these hits, taken them off the beads, squirted them into nitrocellulose, and then flowed fluorescent bacteria over them. And the dried down islands of molecules capture those bacteria. So now we're working hard now to develop these as diagnostic tests that don't involve monoclonal antibodies. Okay, I'm going to switch gears here. I want to talk about something else that we can do with this technology, probably the most important thing, and that is catalysis. So something that's not really appreciated is how nature builds things. Nature builds things by assembling these wonderful, magical things called catalysts, called enzymes, by organizing multiple groups in space and binding metals in many cases, and creating pockets that other molecules can go into, react, and products come out. And the catalyst is not changed by this. And so one catalyst can make billions of product molecules. This is how nature builds us, builds everything in nature. We have done this. We have made individual spiral ligamers that accelerate chemical reactions. Like this one here, we designed it to do what's called a transesterification reaction. It converts this trifluoromethyl vinyl ester into a methyl ester and does multiple turnovers and has, accelerates the reaction about 2,500 fold. This is nothing really to write home about. It's not a very impressive catalyst. Enzymes, biological enzymes, can accelerate reactions trillion fold. And this is something that we need to develop as a species. This is a capability we really need to develop, is to rationally design catalysts. Because if we could do things like create molecules that split water to produce electrons driven by solar energy to produce solar fuels, to lift carbon dioxide back up to alkanes and make solar fuels from sunlight, carbon dioxide, and water. This is something that all green plants do. If we could create stable catalysts that do this, we could create solar fuels, completely green fuels that would solve all of our energy problems. We could make catalysts that would help us make our building blocks for spiral ligament synthesis. so that you could throw into a barrel some proline and a couple of days later get out bis amino acids and drop the cost of the building block synthesis essentially to nothing. You could create catalysts that could selectively modify existing drugs and create new compounds that are not commercially viable right now. All of this could be done if we could create molecules that have pockets in them with metals bound down inside of them, and the pockets control what substrates can get at the metal and react with it. Here's an important application for this. As we age, the proteins in our extracellular matrix react with glucose, sugar, and form these crosslinks called Glucosa Pain. This accumulates through aging, and we have no natural enzymes to clear these out. It's probably responsible for a lot of diseases of aging. It's definitely responsible for wrinkles, tightening of the tissues, the pain you feel in the morning as you get up, as you get older. I'm feeling that. I'm feeling very compelled to work on this project. The problem is that these crosslinks form in the very tight spaces inside of the extracellular network. And even if we did find biological enzymes, like bacterial enzymes that could cleave this group, they won't be able to get into the tight spaces because they're going to be too big. They would also raise a violent immune response if you inject them into people. And there's just a bunch of reasons why this is not going to work very well. So I'm not going to go into this. This is just sort of some background on advanced glycation end products, Glucosa Pain, and how it's very likely related to aging. But what I would propose is that we take the Spiegel total synthesis of Glucosa Pain, which has been published by Dave Spiegel's lab at Yale, and we make a molecule like this that has a fluorescent donor on one end, a quencher on the other end. This is a common thing to do. It's called a molecular beacon. And then develop small spiroligomer-based catalysts that can get into the extracellular matrix and cleave these selectively without cleaving anything else. What these would look like are 3 or 4 spiroligomers tied together into a bundle with a metal bound inside of it and a pocket that would match Glucosa Pain and selectively cleave it. How we would discover this is we would make hundreds of spiroligomers with different metal binding groups on them. We would assemble a DNA encoded library like we do right now for SIMBAs, and we would treat them with the Glucosa Pain molecular beacon, and then look for the ones that generate a fluorescent product, pick out those beads, identify the catalysts, and then study how they work and start to do computational design to improve them. Switching gears again, another thing that we can do with spiroligomers is we can tie them together into little bundles that are like little bricks that are about 2 by 2 by 2 nanometers with lots of control over the functional groups that stick out from them. And we can use cross-linking groups that would let them self-assemble, or using positional assembly, assemble them into more complex structures all the way up to complex machines. So these are just cartoons, but I have been working for the last 20 years to develop software to design these things, and I've gotten this working now. It's sort of like Rosetta, but for unnatural building blocks where you don't have a protein data bank to build from. And CanDo is it's about 1 million lines of code. It's got its own compiler built into it, and it lets you specify building blocks using this sort of syntax here, specify what your foldamer looks like. So this completely describes spiroligomers, our foldamer. To describe amino acids and proteins, you just need 3 lines. You just need 3 entries. But spiroligomers are a lot more complicated. And then what the software does, what CanDo does, is take these descriptions and then run, and I've been doing this over the last couple of weeks, it takes about 1,600 CPU hours to build a set of 1,200 training molecules. And these training molecules are just one building block in a context of a couple of other building blocks and then capped to make a chemically reasonable molecule. And then it does lots of molecular mechanics conformational searching, throwing out things that where you get bad, poor convergence, pulling out all the unique shapes that these training molecules can adopt, and then accumulating all these unique shapes into a sort of a trained database. And then that allows you to build large structures very quickly that we can then search for the structures that can hold groups in particular three-dimensional contexts. So this is going to help us design proteins, spiral ligamers that bind proteins, that create active sites and catalytic sites, and ultimately create machine parts. In 2007, long before we could even put functional groups into these molecules, I wrote part of the roadmap for nanotechnology with the Foresight Institute and the Battelle Corporation and the the guy who did Gateway Computers, the Family Foundation. I'll remember it soon. We had this roadmap for molecular nanotechnology. I wrote a chapter and described how we could do positional assembly with spiral ligament bundles. This is what I would put in these positions, and essentially mimic Stewart platforms with synthetic chemistry, and then use positional synthesis to assemble these into complex structures that could ultimately grow into machines that could build even more complex structures. So that's all I have right now. Just wanted to sort of cap off. We want to develop all these different applications. Right now we're working on this Simba project, but this is a platform technology that could solve a lot of problems. So I'll stop there and be happy to take questions.
Wonderful.
Anybody
Thank you
there?
so much. Yes,
Oh,
we're all here. No.
okay.
We're all here. I think you've definitely stumped some people here. You have a ton of questions already in the chat. And this was wonderful. I'm assuming that we may dive in a little bit deeper into some of these slides. I love that you gave a little bit of a riff of a few things you could be diving deeper on. That was wonderful. Thanks for that. I think maybe we'll start with Logan, with you first, if you want to unmute and ask your question. And from now on, if you want to put your question in the chat or raise your hand, both is fine. Both of.
Great. So I was wondering about the low cost of spiral ligamers, and specifically in comparison to DNA. I was wondering if that figure of, I think the DNA you said $1,000 per gram or something, and then the spiral ligamers were $25 per gram. I was wondering if the figure for the DNA was drawn from DNA that is produced using chemical synthesis, or if it's also including DNA that's produced using biological replication. Okay, so you could, yeah. So would the cost comparison potentially be more similar if you included DNA that you had first chemically synthesized and then amplified using PCR or grown up in bacteria or something?
No, what I'm talking about is chemical synthesis of DNA, and that's just a number that I've heard. If somebody wants to update me with a better number, I just whipped that together this morning, that sort of cost thing. So,
sure.
Look, the big benefit of DNA and proteins is that we have biological machinery, amazing, wonderful catalytic biological machinery that can assemble them. But you get what, so it has that. I do not have that with spiral ligamers.
Sure. Yeah, and I can definitely see the immense benefits of spiral ligamers. I was just curious about how they compared in that respect with that additional sort of qualifier on there. But yeah, thank you. That is very informative. I appreciate that.
Thanks.
Wonderful. We have Priyan next.
I'm not hearing anything.
Priyan, do you want to unmute?
Oh, okay. Yeah, I'll just ask my questions that I put in the chat.
Oh, hey, Corinne. Yeah, I'll pull up. Yes, please go ahead.
So yeah, two questions. One is, what role does simulation play in your work, particularly in the design of active sites? If you do it, kind of what role does it play? What kinds do you use? And then second question is, I noticed the language looked a lot like some dialect of Lisp. How did that come to pass?
So answering them backwards, it's Commonlisp. I'm using that as our sort of programming. Can-do is Commonlisp that interoperates with C++. It's about half C++, half Commonlisp. And we wrote the Commonlisp compiler from scratch.
Wow.
How much do we use simulation? Not much yet. I've been working for 10, 20 years to develop the software to get to the point where we could use it for modeling. We mostly use plastic models up until now, but I plan to use it a lot. I wrote Leap, which is a front end for Amber, which about maybe a quarter of the world's molecular simulations go through every day to set. It's a program that sets up calculations for biomolecular simulations. So I use molecular mechanics. We're using primarily Amber, the Amber molecular force field.
So that's not, I mean, that's fine for maybe guessing a conformation, but it's not so good for designing an active site where all the electronic details have to be just right.
That's what can-do is for.
Excellent. Excellent. Very cool. Wrote a Commonlisp compiler from scratch, huh? That sounds like a lot of time and effort.
Yes, it's open source. It's on the internet, has been for years. It's can-do developers on GitHub.
It has a big fan base, and I think Exciting Creon is definitely a great feat. So congratulations, Chris. Next one up, we have Stefan.
I just want to say I spent years programming Commonlisp, so I'm amused and excited.
Great. I'm really looking for more people to work with us. And if you're interested, get in touch with me. Yeah, we've got a small team that's been working on it. Now it's really, I think, going to open up because we can actually design these molecules with this software.
Awesome. Next one, we have Stefan Boss.
Yeah, thanks for a great talk. So, I mean, I'm interested particularly in the machine side of things. So, I mean, can you incorporate, build these systems so they can display large amplitude controlled motion? So, for example, like sort of a hinge type behavior or rotation of one component relative to another, because obviously that's important for if you're thinking about building like nanomachines.
Yeah, flexibility is easy. Flexibility is something we have to work hard to avoid. So building molecules that have hinges in them is just put a single bond between two rigid units and you have a hinge. We published a molecular actuator that binds metals in open and closed. And we demonstrated that, 12 years ago, or like 10 years ago. It's very straightforward to create things with large amplitude motions. It's difficult to make molecules that are highly structured that you can connect together to get those high amplitude controlled motions.
Yeah, great stuff. And then my other question was with these sort of polymers that you've got all these different sort of units that you can put on, that strikes me as a really cool potential information storage polymer. Have you thought about it from that point of view? And, for example, have you tried reading the sequence of these structures using nanopore sequencing or something like that?
You cannot, because they are complex three-dimensional structures. And a lot of the information is buried in stereochemistry, which is something that is you cannot pull information out of them and sequence them because you can't reduce them to a linear structure once you've assembled them.
Yeah, so sorry, I was just thinking before you assembled them, obviously, but fair enough. Thanks a lot.
Yeah, even before. No, we go in with a sequence that we want. We make that. We demonstrate it has the right mass. We're working on growing crystal structures so we can see their three-dimensional shape. But you can build the three-dimensional shape with a plastic model. And it's complex and rich because of all the stereocenters that can be one of two different configurations. The Simbas we're making have 30 stereocenters in them. And if I make one of those, I think it's almost impossible to figure out what the stereo centers are after the fact. I mean, it's actually a sort of something we could use for intellectual property, protecting intellectual property. Um, the only way you'd really get the structure out is by growing a crystal and then determining a three-dimensional shape of it.
Very cool. All right, thank you. I also just shared the Nanotech Broadband just for giggles here in the chat that you referenced. And then next one, we have Ted Keeler.
I'm not hearing anything. Ted?
Can
You're
you
muted?
unmute? Otherwise, I can
Yes.
read out
Okay,
your
I'm here.
question. Ah,
All right. Yes. Yeah. So I'm excited because the key thing, making an analogy to the semiconductor industry, is to get on a path to profits. So once you
Yes.
can make a product that makes a profit, then banks are willing to lend and things can take off. And so I'm just excited that you have two different potential products here, both the disease diagnostic and the catalyst. So it's just really exciting. I'm just really glad you're doing it.
Yes. And speaking as someone with a $3 million a year burn rate company that's developing this technology, that's something I think about every day. We do have, I mean, I think there's enormous commercial applications. There are enormous commercial applications for this because we can make these molecules now so easily and design them for applications. We have to get out, convince investors, convince people to support this kind of stuff. And that's what we're kind of working on right now. I'm working on a business plan, working on that. We are also driven by the DoD milestones, and
Mhm.
yeah, it's been a bit of a white knuckle ride the last 3 years getting this technology off the ground.
Mhm. Yeah, I'm also impressed with the stability of the things that you've made for disease diagnosis. For the detecting the pathogens. The fact that they
Thank
can
you.
sit
Yeah,
around and last on the shelf for a long time is really good.
right. Thanks. Yeah, that's something that was very important to the Department of Defense.
Yeah, good. Thank you.
Well,
Thanks.
I'm going to drill a little bit more perhaps in like long-term applications and your part there in a second. I just want to make sure that we get to one more question in the chat, which is from Neve, who is driving, and she has two questions. Number one is, how does it differ from Lego SnapChem that, for example, Enamine does? And the second question is, how do you focus on bond types as opposed to DFT type exploratory simulation?
Thank you. The comments, like reading them would be a little self for me. Thank you for reading that out. Okay, so how does it differ from Lego snap chemistry like enamine? So enamine snaps together, it has lots of different building blocks that they snap together through single bonds. And there's rotational flexibility around all of those single bonds. And so you make a molecule that's kind of floppy. It adopts lots of different shapes. And the fundamental principle of structural biology is that structure defines function. If you have a molecule that has a function, it has to have some three-dimensional shape to achieve that function. And so floppy molecules can bind things, but they have to freeze out a lot of their motion to do so. Now, the chemistry that enamine uses to link things together is the same chemistry we use to link segments together, and that we will link modules together with. Um, uh, we basically want to use like efficient, high yielding chemistry that works all the time, reliable chemistry. So we use the same kind of chemistry that enamine uses. It's just the difference is we have rigid building blocks that snap together through pairs of bonds, and enamine has more flexible building blocks that link together through single rotatable bonds. Then how do you focus on bond type as opposed to DFT type exploratory simulation? So I use Amber. I'm an Amber developer. Wrote Leap and been using it for 25 years, 30 years. It's the only way we're going to be able to make lots of different shapes and search conformations quickly we use quantum chemistry to parameterize our models, but our models are sort of a classical model. So using DFT and quantum is something that we use to parameterize them. And we use quantum mechanics to model transition states of reactions when we want to create a catalyst that can stabilize a transition state. So that's a place where we combine quantum mechanics with molecular mechanics. So I'm agnostic. It's just quantum mechanics is very, very slow, scales very poorly, like to third power of the number of electrons you have. So you can't really deal with very large structures. I'm really excited about these new deep learning methods that can predict quantum energies from structures. I'm watching that very closely. It's something I would like to incorporate into CanDo. So I'm agnostic, whatever works, but it takes pretty sophisticated software to properly build molecules, parameterize them properly, run millions of simulations, organize them all, pull results out of it. That's what CanDo is for.
Wonderful. Yeah, I'm particularly, obviously I'm biased because I also run our biotech and longevity group, but I'm particularly really excited about the potential longevity and aging applications. And I just wonder, if you could speak a little bit more to that and just, diving perhaps a little bit more into like what's possible here. Are there any open challenges? What could people potentially get excited about? I know that folks like you don't like to speculate very much, but, making this perhaps a little bit, extra relevant for the longevity community, because I do think this is many of the tools that people in the molecular machines group are building are really relevant for the tools that the folks in our biotech longevity group are seeking. So I'd be really eager to hear you explain a bit more on it.
I think the most of an extremely important application to develop is to be able to rationally design catalysts, to make molecules that can make other molecules, because then you can create the building blocks that you need to make everything from nature's starting materials, which are carbon dioxide, nitrogen, oxygen, water, and sunlight. That's how nature builds everything. We need to develop that capability to make artificial catalysts. This Glucosa Pain application, cleaving Glucosa Pain, is something I'm especially interested in because I'm getting older. I'm feeling the pain every morning. Clock is ticking, and we don't know exactly what is the cause of aging. I mean, I saw a new one a couple of days ago that it's methylation. There's some evidence that methylation patterns in DNA, but fundamentally, any cause of aging is going to come down to chemical bonds that are being formed or broken in the wrong place. And the way to fix that is with catalysts that could get in there and change those chemical bonds back to what you need them to be. So the Glucosa Pain example is just one of those. It definitely builds up with aging. It definitely makes your tissues tighter. By the time you're 80, 90 years old, about 3/4 of the crosslink in your extracellular matrix is Glucosa Pain. We have no way of cleaving that. So I think an idea of making 10 million potential catalysts that have different pockets in them, and then throwing them at a molecular beacon with Glucosa Pain in it, you will find molecules that cleave Glucosa Pain. You could throw a lot of other valuable or like starting materials at that pool of catalysts and pull out a lot of other catalysts that do other reactions, and then figure out how they work, figure out what their shape is, start doing modeling, design better ones, and learn how catalysis works, and then start to rationally design them for specific reactions.
Awesome. Wonderful. Very cool. And could you speak maybe to a few of the other bits on the slides as well, like drawing out a little of a path perhaps for therapeutics as well?
Yeah, so therapeutics. So we have made the individual spiroligmers, the little segments with 3 or 4 functional groups, 10 stereocenters. With just that size molecule, it's about 1.5 kilodaltons in size. We've designed some of those that bind a protein called MDM2. And we published that in 2012. And the compound gets into cells, binds the protein, and actually stabilizes it, makes it, its levels accumulate like 30-fold inside of cells. Those molecules could be drugs. I think it represents a vast new reservoir of therapeutic compounds. We know how to make millions of them and then quickly identify the ones that bind the target protein, figure out what it looks like, how it's bound to the protein, and then start to develop better ones. Medicinal chemistry. The compounds themselves have, I think, good drug properties. They're not going to be chewed up by proteases. They won't raise an immune response. We can make thousands of variants of them and quickly screen them to find better ones. The ones that we've looked at get into cells by passive diffusion. I think this represents an enormous new reservoir of therapeutic compounds. We need to get started on exploring this.
Very cool. Okay, and perhaps a few more words on the sensor bit, because that has so many different applications in like pretty much most of the fields that that we have groups in. So I'm assuming that many of those fields could be interested in that.
So I didn't get the first word. A few more words on
Sensors. Like the
sensors.
top right bit on your slide. If you could just, elaborate a little bit more, like, where do you see potential applications first there? Do you have any ideas already, or?
Yeah, so things like neurotoxins, like shellfish toxins or teratotoxins, small molecules that are made by shellfish or dinoflagellates, or things like polyfluorinated firefighting chemicals, these forever chemicals, PFASs, fentanyl, opioids, lots and lots of molecules. We would like to be able to detect them selectively in very low concentrations. We can make molecules that are like triangular, that point groups into the inside and create pockets. And imagine it's like a little triangular box where you can change the shape of the inside. If you make a shape that's complementary to the molecule you want to bind, like fentanyl, then it will bind that thing. If you attach those triangular molecules to an electrode surface, for like, or like a surface acoustic wave sensor or a chemical field effect transistor, you can turn that sort of traceless or that sensing platform into a selective sensor for that chemical agent. People are trying to do this with antibodies, but antibodies unfold when you put them on surfaces. They don't last very long. They hold their binding site several nanometers away from the electrode surface, while the molecules that we would make would be right down on the sensor surface. There's enormous application here for making long-lived environmental sensors, chemical sensors for all sorts of small molecules.
Awesome, great. Before I go and like literally go down the rabbit hole of like each of these individual bits on there, we have Micah, who had a hand up for a little while with another question.
So this, you may have already answered this, but please answer it again because I might have missed it.
Go ahead.
Proteins have the protein folding problem where we don't know how they fold really unless we use a complicated AI. Does your thing have a similar problem where we don't know what shape it's going to take until after we build it? Or do we have the ability to predict that easily?
Nope. You can take, even someone who hasn't done organic, well, an undergraduate organic chemist can take a plastic molecular modeling set that you get at a bookstore and tell me what any one of my molecules look like, any spiroligomer looks like, in a couple of minutes by building a molecular model of it. They're easy to predict their structures, but they're still complex and rich because the stereochemistry rotates groups in different directions. So it's programmable. So no, we don't have a folding problem with individual segments. We know what they look like. If you link them together with hinges, like we've started to do now, then they have some motions that they can make, but it's quite restricted. And with CANDO, we're going to be able to search through those spaces by just generating, basically using Monte Carlo and a scoring function that scores them for how well they're placing their groups. Like you want to throw out any shapes where two segments occupy the same space, where they overlap in space. So you throw those ones out. So we can use Monte Carlo the same way they do, like David Baker's Rosetta program does, to find shapes that can do specific things. Just search with Monte Carlo and a scoring function that scores how well you are matching the shape that you want to adopt. An example of what we're doing right now is we're putting metal binding groups onto our molecules, onto spiroligomers. I got to stop saying our. I want everybody to use these, but right now we are the only people who make these things. We put metal binding groups on them. We go to the Cambridge crystallographic database. We get crystal structures of metal ligand complexes using the same ligands that we're incorporating into the spiroligomers. And then we look for the molecules that can organize the groups to match the metal binding site that we got from the crystal structure. Then we can change our metal binding site to knock out one sort of group and leave a hole, an open coordination site on the metal where other groups can come in and catalysis could happen. And that's how we could start to rationally design metal catalysts with spiroligomer scaffold around it to create open, empty coordination sites and pockets around those coordination sites to control what can get at the metal.
Awesome.
I
Thanks.
hope I answered your question.
Yes. Yes, definitely.
Yeah. So we have a small folding problem, but it's manageable.
Okay, cool. Well, I would love to, pick up a bit on having you maybe explain, if you want to fill in any more bits on the protein binding, like application that you mentioned here. I know that you've already discussed it a little bit, but in case there's any more detail that you wish to share, that would be wonderful before we get into the longer molecular nanotech goals that are very Foresight and I think of much interest to people here in this group.
Yeah, so the protein binding, I should couch that with, we don't actually know if these are binding proteins selectively yet, because we are screening whole live organisms, and dangerous live organisms I can't tell you about, but we're doing this in a biosafety level 3 lab here at Temple University. We find molecules that capture the organism on the beads, and then we're trying to take those into diagnostic tests. But we don't know what on the organism we are binding. It's kind of the weird way that this whole project got set up. We are now talking to them about targeting a specific protein where we will know what the target is, and we can get a co-crystal structure of the protein with the SIMBA bound to it and figure out how it binds. This is an important thing to do, but we're not doing that thing yet. We are working with live, whole, dangerous, nasty organisms.
Got it. Thanks for the clarification. And then, the last one, and bottom right on the slide that is obviously of big interest to many people here is the molecular nanotech application. And, perhaps that's a little bit further out, I'm not sure in your perception, but what do you think we could get excited for here potentially?
So, what got me into this was Eric Drexler, and the engines of creation and nanosystems. Crap, it's right here. So, I read this stuff. I loved it as a graduate student. That's what got me into this. I really like the stuff that they were talking about, molecular building blocks, less so the positional synthesis with a few atoms. I always thought, right from the beginning, the idea would be to build these building blocks, build larger structures, functionalize them on the outside so that they could mate with each other, so that they would be complementary to each other, but also use chemistry to lock them together. So basically make molecules that are complementary so they can dock, and then have groups that will react when they're docked and lock. So dock and lock was the idea. And be able to first self-assemble complex structures that could be parts of machines that could then be used for positional assembly. I don't think about it too much because I'm really kind of in the weeds with making the chemistry work for the last 20 years, but that's always been the goal. And I think we're near there now because we've synthesized 2.5 million 5 kilodalton macromolecules with lots of functional groups on them. That's what you need to be able to do this. We've scaled the building blocks up to kilogram. That's what you need to be able to do this. So that's where I really want to go with this. It's one of the directions to go with this, to build like a first generation molecular nanotechnology that could then do more controlled chemistry the way Eric was talking from the beginning.
Awesome, thank you. And do you foresee, you know, like along this path, like perhaps this more longer term path, do you foresee any like risks or challenges that, you know, you think we should be at least watching out for as we're building these like pretty sophisticated new structures?
There's always, of course, there's always risks, right? With any technology, that's almost a platitude. I am not worried about some of the early risks that Eric wrote about. Doing things like self-replication is extremely difficult. You know, just get anything off the ground it takes a lot of work. To make something that could actually be dangerous, would take a lot of work. You know, but there's lots of lessons in biology on this. You know, proteins, right? Proteins, 20 amino acids. They're harmless. They're delicious. We eat them for breakfast. But you put them in the right combination and you get botulinum toxin, which is one of the most lethal molecules known. You know, it's 3 units, one that binds a cell surface, another one that translocates across the membrane, and then this catalytic domain that cleaves these snare peptides that prevent acetylcholine, bags of acetylcholine from squirting out of the cell and preventing cells from communicating with each other and causing your brain to fuse. They're the most lethal molecules known. It's not what you put in there, it's how you put it together. So the right combination of spiroligomers, yes, they could be very dangerous. Let's not design those. Let's focus on things that'll really help us. There's so much to do. You know, solar fuels, aging, diseases. We have so many problems. We need better molecules to solve them. I've been working for 20 years to develop these, what I think are better molecules, more programmable molecules, and the software to design them. It's ready to go. I need more people. I need more resources to do this.
Wonderful. That brings me to the last question, because we're on time, which is the standard question of like, if people now get extremely excited about your work, which I think they are, then what can they help to you in particular and your group to really move this work along?
Get in touch with me. And if I don't respond, be persistent, get in touch with me. If you've got software engineering background, we got a big project that we're working on, can do. Commonlisp, C++, using LLVM as our compiler backend. I've got a talk on YouTube about the LLVM can do chemistry compiler stuff. If you're interested in chemistry, come talk to me. If you're interested in investing in this, come talk to me. There is, I mean, right, all that stuff. Right now, unfortunately, it is just us. We are the only people that make it. But I really like other people to make these things, and we're working hard to scale it up and drive the cost down and the complexity down.
Okay, wonderful. Well, I volunteer, at least for everyone in this group, to go through me in case Chris doesn't get back to you in a while, and then I'll join the pinging crowd, and so I can also, I'll be happy to ping you too. Okay, wonderful. Hey, this was really, really amazing, and it's been kind of crazy to see also how much progress has occurred since the last workshop, where we've seen like you know, a partial presentation at least of this, and I'm really excited to see what you were presenting at the upcoming workshop. You're definitely moving pretty fast. So thanks everyone for joining this one, who was here in the seminar, and everyone on YouTube for watching. And yeah, we'll be back pretty soon with the next one. And yeah, get in touch with me if you can't get in touch with Chris. Thanks a lot, Chris. Have a wonderful day, everyone, and I'll see you all very soon.
Yeah, thank you, everyone.